Assalamualaikum everyone. In today's video, we are going to discuss about experiment number 9, and this experiment is about determination of hemoglobin concentration by Sahil's method. So, in this lecture, we are going to discuss the whole experiment of physiology from the first prof. Okay, so we have discussed first 8 experiment in the first video. Now we are going to discuss from 9 to 20 experiments from this video. So now let's start the lecture, and if you don't have to subscribe to my channel, then go and press the subscribe button and like button, and then you have to um, rejoin my video. So thank you so much for subscribing to me. Now let's start the lecture. So first of all, we are going to discuss Wales method, because in this method we are going to detect what the hemoglobin concentration is present in your blood. So what is the Siswell method? In this method, your red blood cell membrane is destroyed by 0 0.1 normal HCl solution. So your 0 0.1 normal HCl solution, first you have to take blood sample, add 0 0.1 normal HCl solution into it, and then what you will going to observe, you will observe a brownish color compound which is acid hematine. Okay. Now RBCs are destroyed by why it is important to destroy the RBCs, because your hemoglobin is present inside the RBCs, so you have to break and destroy your RBCs so that the hemoglobin will become free, and after that you will find out the concentration of hemoglobin. So your hemoglobin is actually going to react with this HCl, and then they form a acid which is called as acid hematine, and it has brownish color. So the normal value of hemoglobin concentration in male is 14 to 18 gram per decimeter cube, and in females its concentration is decreases to 12 to 16 gram per liter. So in the procedure of this experiment, you will take 0 0.1 normal HCl solution in the bottom, and then you will be gradually diluted up to the mark 20 ml in your diluting tube. Diluting tube is very similar to the test tube, just it has a 20 ml grading, and whenever you have to dilute your blood sample till up to the 20 ml, and then after that you will going to perform this experiment. So now you have to prick your finger with the help of sterilized lancet, and after that you have to place the tip of your hemoglobin pipette on the drop of your blood, and your this pipette is going to suck your blood, and you have to suck the blood up to the 20 ml without the formation of air bubble, and after that you have to insert into the diluting tube, and after that you will have to blow out the blood from it. And I mix the blood with HCl, and then you have to shake it for 10 minutes. After that the color of solution will appear brownish. After that you have to add the diluting water into it, drop by drop into the hematine solution, and stir it, and read the scale is given on the distillating tube, and after reading in this scale you will detect what is the concentration of hemoglobin in your blood. So the my concentration of hemoglobin was 10 gram per decimeter cube. So the precaution is that you should not use the first drop of your blood, and there should be no formation of air bubble. All the apparatus should be clean and dry, and the experiment must be performed in the bright light. Okay. So this was the experiment number 9. Now let's go to the experiment number 10. So the experiment number 10 is about the determination of erythrocyte sedimentation rate. Erythrocyte sedimentation rate can be abbreviated as ESR. Okay, so the apparatus needed for this experiment is the Western tube. It is a pipette like, and it has a stand with it. And there is nozzle capillary pipette, which is can be called as posterior pipette. You should need a coagulant, and the normally coagulant you will take is sodium citrate and 3.8% of it. Then you have to take disposable syringes, and some double oxalate and spirit swab. So theory is that first of all you have to see what is the ACR. So ECR is the measure of settling down of RBCs. Okay. So at ECR is the rate at which your RBC are going to down and settle down, or going under the sedimentation. So sedimentation is happening due to the gravity force, because whenever the gravity force is acting on such substances and pulling toward the downward, so therefore the settling of RBCs is occurring. So this term is called as ECR. in a specific coagulant. Anticoagulant should be used because if we don't add the anticoagulant, then our blood should be coagulated. So to prevent this coagulation, we use anticoagulant in it. So there are two methods to find out the ECR. First method is Western method, second is Winthrop method. So in this experiment we are going to discuss Western method because we have Western tube in our lab. So after that, we use Western method for the measurement of erythrocyte sedimentation rate. So the apparatus we need is a Western tube with a stand. Okay, so it is a thick wall glass tube with a length of 30 centimeter. So Western tube length is 30 centimeter, open at the both end. So there is no close end, both end are open, and with the internal bore of 25 millimeter, means the internal diameter of this tube is 25 millimeter and the length is 30 centimeter. And after that, this tube is gradually along the lower 2 to 3 position and have 0.200 millimeter from up to down. So now we discuss Western tube. The tall stand for holding the Western tube is vertically with the rubber pad and at the bottom and screw at the upper end. ठीक है. So ये वेस्टर्न स्टैंड की बात कर रहे हैं कि बहुत सारा टावल्स होल्डिंग वर्टिकली होता है और इसके अपर पार्ट पे एक रबर अटैच होता है, उसके लोअर पार्ट पे सॉरी अपर पार्ट पे स्क्रू है और लोअर पार्ट पे आपका रबर होता है. सो इसकी वजह से हम दोनों एंड्स को जो है वो टाइटली पैक कर देते हैं और उसके बाद जो है वो ब्लड उसके अंदर रहता है. ठीक है. अच्छा, प्रोसीजर की अगर हम बात करते हैं तो टेक 0.5 ml ऑफ 3.8 सोडियम सिट्रेट. तो आपने ये एंटी कोगुलेंट हमारे पास सोडियम सिट्रेट था, तो आपने उसका 0.5 ml लेना है. 3.8% से होना चाहिए. क्लीन करना है और ड्राई करना है. विड्रॉ द 2 ml ऑफ ब्लड फ्रॉम द एंटी क्यूबाइडल वेन. तो आपने 2 ml ब्लड लेना है और उसको फिर इसी एंटी कोगुलेंट के अंदर एसेप्टिक कंडीशंस में मिक्स करना है. रिमूव द नीडल फ्रॉम द syringe आफ्टर ट्रांसफरिंग द ब्लड. Then immediately mix the solution and after that citrate blood is in the western tube. So आपने इस वेस्टर्न ट्यूब के अंदर इस सारे ब्लड को डाल देना है अप टू द मार्क 0. और उसके बाद आपने इसकी दोनों जो एंड्स हैं ओपन एंड होंगे उनको आपने क्लोज करना है अपने इंडेक्स फिंगर के ज़रिए और थंब के ज़रिए. एंड आफ्टर डैट आप इसी स्ट्रैंड के अंदर इसी ट्यूब को वर्टिकली अपराइट जो है वो खड़ा कर देंगे. ठीक है. So press the tube after placing it in the vertical position with the help of screw, and after that note the time after you have to take ESR at the end of 1 hour. Perform the test at the temperature of 22 to 27 degree. A Buffy coat is not included in the reading, so Buffy coat can be used to prepare a blood film of WBC and platelets. So precautions of this is that ESR should be clean and dry, and blood samples should be taken in fasting state. No air bubble formation, no distributing tube, and over the anticoagulant should be used. So these are some precautions. So in the pencil work, again, this experiment is explained that exercise sedimentation rate, the measure of settling of RBCs in an anticoagulant, and two methods, Western and Winthrop methods. Some normal values are given for the man and also for the women and children. So the man below the 50 years age, above the 50-year age has 0.15 concentration of HBC. Then after that, below the 50 years, the concentration is from 10 to 10.20. Okay, so this is the erythrocyte sedimentation rate values. So for the woman who is above the 50 degree is 0.20, and for the woman which is below the 50-year is 10.20, and for the children's its value is 0.20. Okay, so these are the different values which changes according to the gender and according to the age. Now the significance of ESR is very important because it used to has a great diagnostic value. Now let's see some observation and calculations. So ESR in a millimeter of column is first of all it is 9 of the tube, second is the 10 of the tube, and the time which is calculated is after 1 hour. Okay, so after 1 hour, first person ESR is 9 millimeter and second is the 10. So the result is that my blood group has this ECG ESR which is 10.02. So the variation in ECG can occur. It can increases, and the increase in ESR could be a physiological reason and could be a pathological reason. And if it is a physiological condition, then it is due to the high temperature and higher in females. And if it is a pathological condition, then it denoted that you should have some kind of acute infections, anemia, malignancy, and some kind of trauma. So decrease in ESR can be observed, and the reason for this decrease is infectivity, polycythemia, as well as some congestive and cardiac failure diseases. So the result of your ESR should be mentioned, which is 10.2 gram per decimeter cube. So in the experiment number 11, we are going to determine what is visual acuity. So visual acuteness is defined as the acuteness or clearness of the vision. So and it is also defined as it is the power of the eye by which objects are distinguished from the retina image, and which can be appreciated and regarded by its shape and its size. सो क्या मतलब है, अगर मैं आपको उर्दू में इसका मतलब समझाऊँ तो विजुअल अक्यूरेटनेस इसका मतलब ये है कि एक आँख की ये एक ऐसी पावर है जिसकी वजह से आप दो डिफरेंट ऑब्जेक्ट्स जिनका साइज़ भी डिफरेंट है, जिनकी शेप भी डिफरेंट है, आप उसको सो अगर विजुअल एक्टिविटी को देखें तो ये आँख की एक ऐसी पावर है जिसमें आप दो डिफरेंट ऑब्जेक्ट्स को डिफ्रेंशिएट कर रहे हैं, ठीक है। और इसकी दूसरी डेफिनेशन ये है कि आप विजन क्लियरनेस देख रहे हैं अपनी विजन की, कि कितना क्लियर है आपका विजन, आपकी आँख कितना जो है वो सही देख रही है, ठीक है। क्योंकि आपको पता है कि जिन लोगों का सही नहीं होता है विजुअल एक्यूरेट पॉइंट, तो उनको नियर विजन या फार विजन की प्रॉब्लम होती है, फार साइडनेस या नियर फाइडनेस से। सो ये एज के साथ भी ये होता है और कुछ लोगों में एज से पहले भी हो जाता है। सो ये डिपेंड करता है कुछ फैक्टर्स के ऊपर, जैसे कि ये डिपेंड करता है कि रेटिना में कितना शार्प है, या आपका जो है रेटिना में इंटेक्टनेस कितनी है, सेंसिटिव कितना है आपका रेटिना और ब्रेन की जो पार्ट्स हैं। सो ये इन फैक्टर्स के ऊपर डिपेंड भी करता है, ठीक है। सो आपका रेटिना का फोकस जितना ज़्यादा होगा, आपकी आई की पावर उतनी ज़्यादा होगी, ठीक है। इसी तरह अगर रेटिना की फंक्शनिंग और इंटेक्टनेस प्रॉपर होगा तो आपका इमेज भी सही बनेगा। और आपका ब्रेन पे भी डिपेंड करता है, क्योंकि आई से सारे सिग्नल्स ब्रेन में जा रहे होते हैं और ब्रेन ने आगे रेगुलेट करना होता है। सो अगर आपका ब्रेन में कोई पार्ट की सेंसिटिविटी या डिटेक्शन आ गया या कोई डैमेज आ गया, तो उसकी वजह से इस पे ऊपर भी इफेक्ट आ सकता है। इसकी कुछ नॉर्मल वैल्यूज़ हैं, जैसे एक टेस्ट चार्ट हमारे पास दिया जाता है, और इस टेस्ट चार्ट को हम अपने जो पेशेंट है उससे 6 फीट पे, या हम कह सकते हैं 6 मीटर्स और 20 फीट्स के फासले पर होता है। A person can see the letters clearly when he should normally able to see at the distance of 6 meters. So अगर कोई person 6 मीटर के फासले से उन अल्फ़ाबेट्स को clearly observe कर रहा है, so it means कि his eyesight is very clear and correct. And if a person cannot detect it, it means he should wear glasses. So first is the near vision and the far vision. Near vision is the nearest point which you can see from your normal eye and which you can focus on it. So this is called as near vision. It has some definite value. So now let's discuss that. Now this standard value of your near vision is about 25 centimeter and 10 inches. Okay, in the inches its value is 25, and in the centimeter its value is 25. Okay, so the near vision of person is gradually increases or decreases with the help of age. Okay, so if you have this condition, this is called as foresightedness condition. Far vision point is defined as the furthest point at which you can clearly see a object. So the farthest point at which you can see the things clearly, this is called as far vision. And the far vision of person is measured by a chart which is called as Snell's chart. Snell's chart is actually given by Snell's scientist, and this chart is consist of various size of alphabets. For example, in the first row, the alphabet is written with the bold letter, and gradually the size of these letter gradually decreases as you move from up to down. And the person whose eyesight is to detect, then you have to keep this chart 6 meter away from her and him, and then after that you have to ask her to see the chart and to detect the numbers and the alphabets which are written, and after that you will clearly calculate the visual accuracy of that person. So the procedure is that, So for this procedure, the subject is asked to sit at the distance of 6 meters, and after that, the person is tested by the means of Snell's chart. So this chart consists of series of printed alphabets of different sizes, and then this chart has consists of letter in normal size as well as in bold and at the distance of 6 meters. So if a person can read the letter while standing at the distance 6 meters, it means his vision is normal and his vision acuteness is 6 by 6. But if the person cannot detect it, it means his vision acuteness is not 6 by 6. Significance is that it is usually important to determine the visual acuity and the performance and the functioning of your eye. So this is the experiment number 12, and in this experiment we are going to study about the hemocytometer, and the hemocytometer can be called as improved Newbar chamber. Okay, so the hemocytometer is also known as counting chamber, and it was introduced by the Kramer and it in 1805. Okay, so hemocytometer, the second name of hemocytometer is counting chamber, and the scientist who discovered it was the Kramer, and in 1805 he discovered it. And a hemocytometer is also modified by two scientists, which are Thoma and Newbar. So therefore it is called as Newbar chamber because Newbar improved it. And so now it is used to count the various type of blood cells. For example, your RBCs, your WBCs, your eosinophils, your platelets, all type of the blood cells which are present, they can be calculated or counted by this hemocytometer or improved Newbar chamber. So for now, let's discuss the parts of hemocytometer. So it first consists of counting chamber. So the counting chamber is a special slide which is H-shaped, and it has some trough and crest in it. So the platform is exactly 0.1 millimeter deep below, and the cover slip is going to cover it, and on the ridge, and the space is below it is about 0.1 millimeter. So now the height is created below the cover slip is on the central platform. There are small number of scales which are present in the area of 3 millimeter and 3 millimeter long and 3 millimeter wide. So each consists of 9 large scales and has one. These 9 large scales have 1 square millimeter in each. The 4 large scales are represented at the 4 corners. So these are used to count the WBCs, and these 4 corners are divided into 16 small squares. So now these 16 small squares are further divided up to and they consist of approximately 80 small squares. Then now the squares are represented at the center. They are used for the counting or RBCs. These squares are further divided into the 25 medium size of squares, and their volume is 1 over 250 millimeter cube. So these scales are further divided into having arm of 1 over 20 millimeter, and the area is this and the volume is that. So the RBC are counted in 5 medium-sized scales, and these scales are given the name of R1, R2, R3, R4, and R5. So what is the RB pipette? So these are the two types of pipette which are used in this hemocytometer method. First is the RBC method pipette, and second in the WBC pipette. So RBC pipette is a glass tube which has a capillary tube in it, and it has a red bead in the bulb portion, and a small stem is connected with this bulb. Now the capillary stem of this pipette consists of some readings. For example, it consists of a reading of 0.5, 1.0, and 111. So these readings are actually useful in detection but where is the section of it. So the stem has approximately one unit. Okay, it is divided into two readings, 0.5 and 0.1 reading. Then after that, the bulb portion contain, and the bulb portion of red blood cell pipette is consist of 100 units. So after 0.1 reading, the reading of 101 came. Okay, so the total units which are present in RBC pipette is 100 units. Okay, so the bead which is red color bead is also present, and the diluting fluid mixed with the blood, and after that it is put in this pipette. There is a WBC pipette, and WBC pipette is very similar in the shape of size with the RBC. Just the little difference that its bulb size is very small, and there is the difference in the marking. For example, it contains a marking of 0.5, 1.0, and 0.11, and it contains of total 11 units, and the capillary stem is consist of 0.1 and 0, 0.5 and zero point 0.1 marking, while the bulb unit is just divided into the 10 units, while in the WBC RBC pipette it divided into the 100 units. So the total units which are